Can you hear me? Can you hear me? Okay, good. Okay, I am going to today I am going to give you help to structured first question. You know, structured paper first question. Now I'm not going to today analyze one by one. You know, it is fully bio question, biology questions. Then in our syllabus, how many bio lessons? We have only two: biomolecules and cell structures. Okay, I hope to do biomolecule seminar, but unfortunately we couldn't. Okay, if we have time, let's try to complete biomolecule. I think you don't need, you already complete the biomolecule, but in cell structure lesson, cell parts, the tissue types of plant, stem, root, leaf part, I think you're not familiar still not well familiar. Therefore, I'm going to explain a little bit about plant, stem, root, and leaf structures. Okay, it is actually just as a very simple story. Try to understand as a story. Okay. Okay, it is like this. I'm going to go for less tute. Get your cell structure lesson. Tute, cell structure tute. Cell structure tute. Cell structure tute. Okay, this one. Okay, in lesson they given first the stem structure of dicot and monocot stem, but I am going to starting with root. I'm going to starting with root, then you can get the full idea about plant structures. Okay. Roots, plant types. I'm not going to explain from the beginning. Now you already know plant types mainly two: monocot and dicot. Monocot. Yeah, in the view, okay, what's the main first and dicot? Monocotyledon and dicotyledons. What's the main difference? Okay, I have given in page number 32. It has only one cotyledon. That means monocotyledon plants, only one cotyledon in the seed. Like this. It is only one cotyledon. If you got monocotyledon plant, it is two cotyledons. Like this. That's the main difference. Monocotyledon and dicotyledon. I have monocot plant and dicot plant simply. Okay, that's the main difference between monocot and dicot. And I have given root system in monocot fibrous root system. In dicot, it is a taproot system. Taproot in long root. Stem in monocot stem don't have like coconut tree, like don't have branches in the middle of the stem, but in dicot contain branches. And monocot uniform, the stem uniform thickness. Clearly, I have given. I think you already followed. Non-uniform thickness in dicot plant, the stem. the leaf, if you got leaf in monocot plants like coconut, it is parallel, right? It is parallel like this leaf, parallel. And leaf attachment and floral types in flowers of monocotyledon plants, number of petals, tree, mostly tree. But dicotyledon plant, it is number of petals, it is 4 to 5 in dicotyledon plants. Okay, they are the main difference between monocot and dicot. I think you have full idea. Okay, now I am going to explain not about monocot and dicot plants. I am going to explain the difference between structures of root, stem, and leaf of monocot and dicot plants. Okay, I'm going to starting with root. I'm going to starting with plant root, monocot plant root. No, let's start from dicot. It is easy. Okay, then I'm going to start from dicot plant root. Okay, take a paper and draw these diagrams clearly. Then you can look attach kara. Then you can remember every parts and their functions. Okay, clear? Dicot plants. Dicot root system, root structure. Right, dicot root structure. Hari, andi mate. Okay. Ada mate තියාගන්න ඕනේ මේක. Shape recognize. Okay, then okay, let's start. Draw a circle like this. Draw a circle. Draw a circle like this. Then draw another circle. Inside of this circle, draw another circle. Okay, මෙතනින් මෙහෙම එන්න ඕනේ මේ size එකට. I think it is a technical issue. Like this. Okay, let's draw. If you only watching, you can't remember these things. Let's watch. Let's draw. Okay. Okay, then okay. Remember, they are root hair. Root hair. Root hair. They are root hair. Root hair mean just like hair actually. They important for absorb water and minerals. Absorb water and minerals. Okay, then this gap we call the gap, like it is a layer, that cell layer, tissue layer we called as epidermis. 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 Then draw another circle like this. Okay, that's name is then another circle inside that. Make a හරියට recognize කරන්න මේක inside තියෙන්න ඕනේ. මේ වගේ like this. Okay, then there is a star inside that, like this. There is a star. Okay, that's the structure. That's the structure. Okay, very simple. Okay, then I will name this. Then this layer, tissue layer, we called as cortex. It is cortex. 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 Okay, that we called as endodermis. Okay, you need to remember this. You know, all in model paper also this is a question, no? In model paper also this is a question, and a lot of past papers also the first question given uh, label few parts. Label few parts. Label few parts. Endodermis. TE, endodermis. And inside that, this cell layer that we called as pericycle. 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 Finally, this part, this may spaces, that we called as xylem, that we called as, sorry, phloem. Phloem. Inside that, they are called as xylem. Xylem. Okay, that's the structure of dicot root structure. Dicot root. Okay, dicot root. Are you complete? Dicot root. Dicot root. This inside is the xylem. This is the phloem. This part is the phloem. This inside layer is the xylem. Okay, do you know what's the difference between xylem and phloem? The xylem and phloem is which tissues? They are complex permanent tissues. I think you can remember. Complex permanent tissues. Xylem and phloem. What is the main function of xylem? What's the main function of xylem? Others, quickly. What's the main function of xylem? Yes. Transport the water. Yes. Transport water and minerals mainly. Minerals mean nutritions. Nutritions. Transport by this xylem. You can understand the xylem is the main middle structure. In root systems, root decay, the main function is water absorption, right? Water absorption. Then this is the xylem. This is the xylem. This is the xylem. Around that, these parts are, they are this green color. They are phloem. Green color phloem. Okay. The outer layer of this xylem and phloem we called as pericycle. Pericycle, remember, it is pericycle. Pericycle. Outside of these endoderms, then this layer is cortex. The outer tissue layer is epidermis and root hair. Root hair. They are the main parts of dicot root structure. Are you complete? Please send me message if you complete. Okay, let's move into monocot root structure. Monocot root structure. Monocot plant root structure. Root structure. Monocot root structure. Monocot root structure. Okay, monocot root structure mean again take another paper, take another A4 and draw look what on the draw a circle. Draw a circle. Then again draw another circle inside of this. Again, you need to maintain the same gap. And same. It is root here. Root here. Root here. It's root here. Root here. Then what is this? What is this cell layer that we called as epidermis? Epidermis. 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 Same, okay. Same as dicot. Epidermis. Epidermis. Then draw again pericycle. No, endodermis. Endodermis. Then pericycle. Now this is cortex. 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 Endodermis. Endodermis. This is pericycle. 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 The main difference of this monocot and dicot up to now it is same. Methane same. Now compare with the dicot one previous page. See, up to now it is same. Up to pericycle it is same. Now, but the internal structure of pericycle it is different. It is different in monocot stem. Okay, understand the difference. It is not actually perfect organized. No, not differentiated. Oh, give me a second. Sorry. Okay, I told you up to pericycle. Up to pericycle. Oh, what happened? Up to pericycle, all structures are same monocot and dicot. We discussing about root. Okay. Plant initial point we start karla. This is a root structure in monocot plant. In dicot plant, it is perfect differentiated, this xylem and phloem. See, it is perfectly differentiated. It is differently available, but organized. But in monocot structure, it is not perfect organized, but not perfect differentiated. They are the phloems. They are the phloems. Phloem tissue. Then they are the xylems. Okay, they are xylems. Okay, that's all. And other thing, in monocot plant, there is parenchyma cell core එකක් තියෙනවා. There is a parenchyma cell core. Parenchyma cell core. Parenchyma cell core. It is actually primary root. Okay, primary root, length increasing root. I think you can you already know primary and secondary growth. Okay, now I will explain the stem well. Okay, it is primary root. Primary root means normal plant root structure. Okay, then that is a parenchyma cell core, mean parenchyma cell core. COR. Core. It is a cell collection in parenchyma. Parenchyma mean immature tissue structure. Immature tissue structure in the center of this pericycle. Center of this pericycle. Okay, that's all. Yes, cambium tissue, definitely I will explain. Cambium tissue. In next part. Yeah, it's a beautiful story. Okay, root okay. You need to draw. After class, you can again draw these cells, structures, again label. Practice, again draw and practice, draw and practice. Okay, that's the method. Then you can't by heart them. You have to practice. Andina label karan, andina label karan. That's the method. Okay. Normally cell core mean it is a immature cell structure here, parenchyma. When the root matured, this parenchyma cell convert into sclerenchyma and sclerenchyma mainly, not sclerenchyma, parenchyma, and second one what? Corenchyma. Yes, corenchyma. Then it is important to the strength mainly, not main function. It is important to strength of the root. Okay, let's move to the stem, right? Stem is very beautiful story. Okay. Okay, I'm going to move into dicot stem structure. Dicot stem primary growth normal structure. This is stem structure. Dicot plant stem structure. Dicot stem structure. Okay, dicot stem. Okay, draw again a circle. Draw a circle again. Okay, then again draw a circle inside this. Okay. Okay, in dicot stem don't have, it is same as dicot root. The outer layer is epidermis. This structure region is tissue region is epidermis. It is mainly important for the protection of the plant. Protection of the plant. Epidermis. Epidermis. Epidermis, that is important for protection purpose. Protection purpose of the plant. Protection purpose. Protect the internal structure. Protect internal structure. And the inside the root, that is cortex. You already know it is cortex. It is same as root. It's same as root. It's same as root. Same as root. Okay. Then inside this cortex, okay, draw clearly these shapes. It's not triangles. Actually, this is actual shape. I can't draw all same as this. It's not triangle. It is a, okay, that's the actual shape. Normally it is it can be that we called as vascular bundles. This number of bundles can be 8 to, it is only 7, no, not enough. Let's 8 to 13, that can be the number of vascular bundles can be changed according to the plant size. Number of vascular bundles. Okay, please draw 8 to 8, 10, 13 between value. Okay, now I have drawn 8. Okay, that's a small plant stem. According to the plant stem size, the number of vascular bundle can be changed. It is normally changed 8 to 13, 14, it is changed. Vascular bundles. Vascular bundles. Okay. They are the cambium tissues. Cambium tissues. Okay, what are the meaning of these cambium tissues? Cambium tissues, you already know meristematic tissues, apical meristems, intercalary meristems. Apical and intercalary meristems, they are simple tissues, simple permanent tissues. Do you remember? They are made with same cell size, same type of cells. Parenchyma, parenchyma, sclerenchyma. Yes, they are. And meristem tissues. Sorry, parenchyma, parenchyma, sclerenchyma are the simple permanent tissues, and meristem tissues. Meristem tissues mainly three types: apical meristem, intercalary meristem, and partial meristem. Lateral meristem. Sorry, lateral meristem. Intercalary meristem, lateral meristem. Okay, this meristem is not the cambium. Cambium mean they are plant tissues. No? Cambium tissues are, they are dividing and producing new cells. That's the main function of these cambium tissues. Cambium tissues are type of tissue that is always dividing and produce new tissues. I got Arshad? They are always dividing and produce new cells. That's the main purpose of cambium tissues. Okay, now that we called as in with the middle of this vascular bundle, this full bundle we called as vascular bundle. Understand? Vascular bundle. Vascular bundle. The full bundle we called as vascular bundle. The middle of this vascular bundle, it is cambium tissues. Okay, cambium tissues. Now later I will explain. It is mainly two types. I will explain what are the two types cambium tissues. Okay. Okay, then what is the first region? It is epidermis. Then inside that, cortex. On the cortex, between cortex, it is contained vascular bundle, and they are cambium tissues. Cambium tissues. Cambium tissues. Cambium tissues. Cambium tissues. Cambium tissues. Okay, this cambium tissue, and what is this as a cap? What is this? Do you remember? What is this as a cap? What is this? That is a cell tissue layer that we call as sclerenchyma tissue layer. Sclerenchyma. Sclerenchyma. What is the actual name? Sclerenchyma. Yeah, sclerenchyma tissues. Sclerenchyma. That's a dead cell region. Dead cell region. Sclerenchyma. That is important for the strength of the stem. That's sclerenchyma cell tissue region. region. Okay, now again revise. What are the parts? The final part is epidermis. Inside there is cortex. Inside cortex, 8 to 13 vascular bundles. Vascular bundles. Middle of these vascular bundles, cambium tissues. Cambium tissue. The edge of this vascular bundles, they are sclerenchyma tissues. Sclerenchyma tissues. Then what is this? What is this remain? Okay, always outside of, I will mark on this one for easy understand. Outside of this cambium tissue, cambium making outside, it is always phloem. Remember, don't change this. It is phloem tissue. Inside of the cambium, it is xylem. Inside of the, that is primary. Okay, that is not the secondary, that is primary. It is phloem or xylem or secondary, no, primary xylem. Primary xylem and primary phloem. Okay, no need to write primary, secondary. It is just xylem and phloem. But it is primary xylem and phloem. Don't take confused. It is xylem and phloem primary. It is still primary structure of the stem. It is not the secondary structure. Secondary structure is a different story. Secondary structure, normally primary structure mean at the initial part of the plant. Normally when the plant is increasing the height, increasing the height by apical meristems and intercalary meristems. You already know apical meristems increase the height of buds area and shoot apical meristems. In intercalary meristems increase in the length of between two what we called two rounds, what we call them nodes, two nodes, internodes. Increasing the intercalary meristems, increasing the length of two nodes, increasing the length. Okay, intercalary meristems. Actually, this secondary growth mean before secondary growth mean actually the purpose of lateral meristem. Okay, what is the purpose of lateral meristem? Lateral meristem increasing the diameter. Yeah, that is the secondary growth. The dicot plant increasing the diameter is the secondary growth. In secondary growth, it is primary xylem and phloem converting to secondary xylem and phloem. That I will explain later. Okay, now I clear these all parts. Okay, that's the basic structure of this dicot plant stem. Okay, please complete this. Dicot plant stem. Okay, draw it clearly. Okay, in the tute I have given clearly diagram. These vascular bundles beautifully organized with same distance. Okay, yeah, like this. They are beautifully arranged. See. Okay, complete. Finish, Deneth. These all names you need to remember. Functions, okay, functions you can understand: cortex, epidermis. Okay, primary xylem. This is parenchyma tissue region for strength of the plant. The cambium. What is the main function of cambium? To produce new cells. Produce new cells. Okay. Okay. Now, when this is the initial primary structure of plant tissue. Sorry, plant state. Plant state. Okay. When the plant matured, what happened? This when the plant mature, these meristem tissues, cambium tissues, it is cambium tissues start to produce new cells. I told the main function of cambium tissues produce new cells and growing of the plants by adding new cells. Okay. In this cambium tissue, what they're doing, they start to produce new cells when the plant mature. When the plant matured, they start to produce new cells. Okay, then what happened? Actually, this cambium tissue we called as inside this vascular bundle that we called actual name is uh, intracalary meristems. Sorry, intracalary cambium, not meristem. Meristem and don't mix this meristem and cambium. Intra mean inside vascular bundle. Intracalary cambium. Intracalary cambium. This is intra. This is intra. Intra. Okay, between this that we called as intercalary cambium. Intercalary cambium. Intercalary cambium. Intercalary cambium. Okay, now the most important thing. This is the most important thing. This is the most important thing. This intercalary cambium and intracalary cambium. Okay, these all cambium tissues add together and produce a cell region like a circle like this. It is produce new cells and it is developed as a circle. It is like a band like this. Like a band. Okay, that we called as, what's the name? Vascular cambium. This ring is called as vascular cambium. Vascular cambium. Okay, for better understand, okay, let's draw again the secondary growth of plant stem. Okay, up to now, that is a primary growth of plant stem. Primary growth of plant stem. Okay. Then let's again go to another diagram for understand the secondary growth of dicot plant stem. Okay, I'm going to draw again the secondary growth. Okay, secondary growth. Okay, this is the epidermis, and inside that the cortex, and they are the vascular bundles. Vascular bundles. They are in same size. Okay, in my drawings, it's not same size. 3, 6, 8. Okay, they are vascular bundles inside the cortex. Okay, they are vascular bundles, and they are the cambium, intracardiac cambium, intra. Cambium tissues. Okay, then other names are same. This is I think you can remember epidermis, cortex, then this intracambium and intercambium. Inter mean between that. Intercambium connect together and produce as a ring structure. It is produce a ring. Maybe a ring that we called as vascular cambium. That we called as vascular cambium. Vascular cambium. Vascular cambium. Vascular cambium. Then you already know this is xylem. This is now we call that is primary xylem, because it is still primary structure, not secondary. Tama primary xylem. Sorry, phloem. Outside phloem, inside primary xylem. Primary xylem. Primary xylem. And inside there is another cell layer forming that we call as pith. Okay, that is the when the plant stem become matured, little bit matured, the structure change like this. Then what is a new cell is forming? Vascular cambium is forming. Vascular cambium is forming. Vascular cambium is forming. Okay, in the lesson I told, in the secondary development, secondary growth of plant stem, what are the new tissues forming? Vascular cambium and cork cambium. They are the main tissue. Always exam asking. In the secondary growth of plant stem, which tissues are forming? Vascular cambium tissue and cork cambium tissues. Okay, now you already know vascular cambium. Vascular cambium, what is the purpose? What's the purpose of this vascular cambium? Okay, now vascular cambium. Okay, that's a new cell tissue is forming, vascular cambium. This inter and intracambium connect together, then produce like a ring structure that we call as vascular cambium. Vascular cambium. Okay, next, it is not the secondary growth, it is only ready for the secondary growth. Understand, it is ready for the secondary growth. Okay, then it is start to secondary growth. Secondary growth means simply it's try to increase diameter of the plant stem. Yeah, mahat wenna hada. Try to increase the diameter. Okay, then what they doing? In the final stage of the secondary growth, now they are increase the diameter. I explaining how to increase the diameter. How to increase the diameter? How to increase the diameter? Then this is the epidermis. Epidermis. Okay, I'll get names from this end. Epidermis. Epidermis. Sorry, I will explain about epidermis later. Epidermis I will explain later. Think this is the produced ring. This is a produced ring. This is a produced ring. Ring. This is the most important diagram. Okay, a lot of questions coming from this. Why it is not there? Okay, this is a produced ring now. Ring. This ring. Okay, this vascular cambium. This is vascular cambium. This is vascular cambium. Vascular cambium. From this vascular cambium, it is, I told cambium tissue always produce new cells, right? New cells produce into inside. What is a cell structure? It is xylem or phloem. Inside, send me, Arshad. Vascular cambium inside xylem or phloem. May inside the xylem or phloem. Xylem. Inside always xylem. Very important. Inside xylem, no. Then it is grow as new cells. It is grow new cells and produce new xylem structure that we called as secondary xylem. That we called as in the main structure that we called as secondary xylem. It is just as a ring, okay? That is the secondary xylem. That's the most important cell structure in secondary growth. In after secondary growth, that we call secondary growth can the diameter of the plant increasing. The diameter increasing due to this reason. It is produce vascular cambium produce new tissues and increase the xylem thickness. Xylem thickness. Then that is the secondary. Understand very well. Secondary xylem. Secondary xylem. Then what is the outside? What is outside, Arshad? Outside of vascular cambium? It is phloem or xylem. It is phloem. Then it is produce another ring. Another ring. That we call, I will draw it in the another color. I will write in. This one called as secondary phloem. This is the secondary phloem. Secondary phloem. Secondary phloem. Okay, please draw very clearly. Look at the gun and name this. Okay, it is just same as a very simple story. Okay, it is a simple story. Okay, then what happened to the primary and secondary xylems and phloems? They already contain. They already contain, but they are very small now. They are very small now. What තියෙනවා තාම. මෙන්න මෙහිමයි xylem තියෙන්නේ. Phloem and xylem. Phloem and xylem. Phloem and xylem. Now you can see due to this new cell development, new cell development, due to this new cell development, the thickness, so not thickness, the diameter of the plant stem is increasing. The diameter of the plant stem is increasing. Okay, they are the, okay, now they are primary phloem. Primary phloem මේ විදියට තියෙනවා, මෙතනම තියෙනවා. Okay, very clearly you need to draw this one. Okay, please complete that up to now, then I will explain the final story. Final story. Okay, now I told in the lesson, in the secondary growth of plant stem, there are two tissues are forming. What are the tissues? Vascule cambium and cork cambium. But still you don't know cork cambium, no? Okay, please note, complete up to now. It is a very beautiful story. Try to remember this as a story. Secondary growth of plant stem. Complete. Complete. Quickly, quickly. Okay, now I told in the secondary growth, due to increase the thickness of these tissues, what happened? This epidermis, I mean, in primary structure, epidermis is breakdown. Okay, that's another very important thing. Okay, you need to remember, due to increase the thickness of these xylem and phloems, as a secondary primary and secondary xylem and phloems, the epidermis, the outer protective layer is breakdown. Outer protective layer, epidermis is breakdown. Breakdown vela, මෙන්න මෙතන new cell, new tissue is forming, then it is required new tissue for protection. Then it is producing new tissue layer that we called as cork cambium. That is the cork cambium. Cork cambium. Okay, very important. Okay, it is called as coke cambium. Metan coke cambium make a form now. Okay, coke cambium, it is producing coke tissue. It is the coke. Coke. C-O-R-K. C-O-R-K. Coke tissue. Okay. The cambium is always dividing and producing new cells and tissues. Then cambium, coke cambium, what they're doing, they're producing new cells and produce coke outside. Then it is very important for the protection of this structure, the stem. Okay, because when increase the thickness of this internal structure, what happened? The epidermis is breakdown. Epidermis breakdown vela, it is produce coke in the outer layer of the. Then it is mainly contain sclerenchyma. Coke mainly the sclerenchyma, dead cells. Dead cells. Okay, that's also frequent asking MCQ question. Okay, any doubt about secondary growth of plant stem? Okay. Okay, you don't need to practice to draw this structure. In exam, not asking to draw these structures, but they always asking label. Wood cambium. Where is it given? Wood. Where it is given wood cambium? Wood cambium and cellular cambium. Wait, let me check. I'm not sure. No. Wood cambium mean vascular one. It is the same word. Same word for vascular cambium. Adish. Vascular eකට තමයි wood cambium. The outer one is coke cambium. It is the outer layer is produced. Then it is important. I have given in the tube. The good tube. The coke cambium. Outer cortex produce coke towards outside. These are important for protect against water loss and pathogens. For protection, it is very important. Okay, then after discussion, you can read the note, then you can get full idea. Okay, okay, that's all about stems. Okay, let's move into final plant part. The final plant part. The leaf. Okay, this is the leaf. Okay, it is very difficult to draw this. Okay, the outer layer of the leaf is cuticle. The outer layer is cuticle. It is like a wax layer. Okay, it is important for protect plants from water loss. Okay, it is a cuticle layer. It is like a wax layer. Then epidermis, upper epidermis, and there are lower epidermis. Then it is a protective, and it is contains stomata. That's mean holes. They are important for transpiration. And they are like a, this is a parenchyma cell type that we call as palisade cells. Like linear shape. That's why it is contained mainly parenchyma cells, parenchyma tissues. Okay, these names you need to remember. And this layer, parenchyma, mean you already know it's contain chloroplast. They are photosynthesis. It is important for the photosynthesis purpose. And they are the xylems. Xylems important for water mineral transportation. They are the xylems. You can clearly understand the middle one. Mainly middle contain xylems. Outside layer is phloem. They are phloems. Okay, you can go with the tube. And spongy cells, they are a little bit matured now. They are parenchyma, and parenchyma both. They are also contain chloroplast. Then they are important for the photosynthesis. Okay, they are the main structures. Please remember these all names: cuticle, upper epidermis, palisade cells, bundle. This is the protective layer we call as bundle shape. And xylem phloems, spongy cells, stomata, and lower epidermis. Okay. Okay, I have given every functions also. Yeah, plant I have given. About leaf I have given full story. You can, how to photosynthesis, everything I have given. Okay, that's all about plants. Okay, I will get another time, 1 hour time, and I will explain about animals also. I will explain. Then the cell structure, every part we covered. Okay, tomorrow I think we can get 1 hour time, no? 10 to 11. tomorrow Thursday, no? 10 to 11 can we get? Okay, I'm getting small time periods from your time per day because I think you have a lot of works now. That's why I'm getting very small time. Now today I got only 1 hour and 20 minutes. Okay, can we get today 1 hour? Sorry, tomorrow, not today. 10 to 11. Okay, let's do tomorrow 10 to 11 about animals. Okay, I think today you got the full story behind the primary growth of stem, secondary growth of stem. Okay, practice again. One more time you need to practice. Then you can remember this. Take as a story and draw these pictures clearly and display in your room. Medical attach karala tiyaga with these names. Very simple, right? Same as. Okay, remember as a story and diagrams with diagrams. Okay, that's all for today. Let's meet tomorrow 10 p.m. Okay, good night. Practice one more time.